Good afternoon, my name is Robert Harris. I go to Lane Tech High School. I'm going to be a senior.
And my name is Jocelyn Ramirez, and I'm going to be an incoming junior.
What we're presenting to you today is genetic diversity of Oenothera harringtonii and differing habitats. Some general background: Oenothera harringtonii is also known as the Colorado Springs evening primrose. It is endemic to southeastern Colorado, which means that it's only found in that area. As well as being endemic, it is also a rare plant. It's pollinated primarily by the hawk moth and a barrage of solitary bees. There's 2 known species of hawk moth that pollinate Oenothera. Some basic ecology: it grows typically at elevations at 1,400 meters to 2,000 meters above sea level. It prefers silty clay-type soil, which is characteristic of where it's found. It's found in open grasslands and shortgrass prairies. It also grows on gentle slopes or typically flat ground areas. Oenothera harringtonii is self-incompatible, which means that the pollen from one flower cannot pollinate that same flower, which makes it an obligate outbreeder. And this means that it's heavily dependent on its pollinators for cross-pollination. Like I said, it's pollinated primarily by the hawk moth, and they're attracted to the strong fragrance of the flowers, as well as the nectar that Oenothera provides. They can travel up to 30 kilometers each night, which is roughly 20 miles. And since they can travel so far, this contributes greatly to long-distance gene flow.
Alright, during our 8 weeks at the lab, the question we further wanted to explore is how does habitat location affect the genetic diversity of Oenothera harringtonii? What we hypothesized is that the subdivided populations would be less genetically diverse than those found in a more natural habitat, simply because we thought that Oenothera harringtonii's pollinators would be disturbed by traffic or by the road. And as Robert said before, they could only fly 30 kilometers a day. As you can see, the ones found in the left are subdivided ones near to the road, and the ones to your right are more natural habitats. Alright, our experimental procedure was based on microsatellite analysis, and basically we used this to determine genetic diversity. We started off with 10 samples, and we used the CTAB method to extract the DNA from. We then ran PCR, we sequenced them, and we did data analysis. As I said before, the method we used to extract the DNA was the CTAB method, simply because it's more affordable than the kit extractions and gives us a larger concentration of DNA. It's commonly used in plant genetics because it could be used for both wet and dry samples, and as seen in the picture in the far right, some materials we used was a CTAB buffer and mercaptanol to lyse the cell and to spill its contents out. We then used ammonium acetate and isopropanol to do a series of washes, and last but not least, we used the TE buffer to get our pure contents of DNA. PCR simply means polymerase chain reaction. It's used to amplify useful portions of DNA and RNA some scientists may want to look at. This process is more efficient than using bacteria to amplify the DNA and takes a lot less time, and consists of 3 main steps. The first step is denaturation. As you can see in the picture, the DNA is heated to 94 degrees Celsius, and what it does is separates the 2 strands, forming one single strand of molecule. The second step is annealing, and in this process, the DNA is cooled back to its normal temperature, and primers are added in to separate the specific section of DNA, and 2 strands come out. The third step is extension. DNA polymerase makes a temporary strand until enzyme sends nucleotides to pair them up. The whole process could then be repeated again and again and again.
Sequencing and analysis. Once the DNA is extracted, before we actually do any analysis, we typically use a machine called a nanodrop. And what the nanodrop does is measures the concentration of DNA in the sample, and that's a picture of it. What it does is it shoots a laser through the sample and returns the frequencies of everything in the sample. As an analysis tool, we use gel electrophoresis as well as a sequencer. Both methods accomplish the same task. What gel electrophoresis is it uses an agarose gel to sort of measure the length of each DNA fragment. We load the DNA into the gels and then we run a current over it, and depending how far it migrates, this shows us how long each fragment is. And the sequencer does the same thing, except it just uses more high-tech equipment. The results: the bold underlined populations are our natural populations, and the 2 at the bottom, Munson and Walsenberg, are roadside subdivided populations. The first column, N, is the sampling size, and this is just how many individuals were sampled within that population. If you look at NA stands for average variation found at each marker. Like Jocelyn introduced, we use microsatellite analysis. And what microsatellites are just repeating sequences in the DNA. And every time there's a change or a mutation in that sequence, it changes the length of the fragment, and then it's just recorded and averages out there. HE stands for genetic diversity. This is actually used as more of a comparison tool. It's not a definite tool to say one species has a lot of diversity. It's more to say population A has more diversity than population B. To visualize this more, the green bar is HE genetic diversity, and blue is the average variation found in each marker, but I want to focus on genetic diversity. You can see Cone Mountain and David's Canyon have greater diversity than Munson and Walsenburg, and this is actually what we initially hypothesized. What this shows us is, again, the names of the populations and the sampling size, and F stands for the inbreeding coefficient. And what this tells us is just how inbred the population is. More visual, you can see actually Cone Mountain and David's Canyon have greater inbreeding than Munson and Walsenburg, and this is actually sort of what we expected because of the spacing of the populations. There was no even spacing. The more natural areas were more secluded and farther apart.
All right, and this data chart, it basically shows the genetic distance from one another. Some numbers we should look at is Munson and Cone Mountain. They have the least genetic distance from one another, meaning they're closely related, although one's in a more subdivided area and the other one's in a natural habitat. The second number is Walsenburg and David's Canyon, which is 0.235. This has the most genetic distance, meaning they have the most unique genetics. For future considerations, if we want to conserve these plants, we should conserve Walsenburg and David's Canyon because the more unique the DNA is, the better. In conclusion, based on our data, we found out that the natural habitats have a greater genetic diversity than those found in a subdivided area, which supports our initial hypothesis. And although the roadside populations were less diverse, it showed us that they inbred less than the natural habitat populations. This was expected because the spacings of the populations. The roadside populations were closer together while the natural populations were more secluded. For future considerations, we would like to add more populations and more individuals, and we would like to have them more evenly spaced out. We would also want to add more variables because more variables mean a better trend. Some variables we would like to add are different levels of fragmentation, pollution sources, and whether the plant is viable or not. In the future, we would like to look for other features of Oenothera harringtonii, such as floral anatomy, scent, and size.
Some conservation considerations. Since Oenothera is a rare and endemic plant, we think more effort should be put towards conserving it. Some methods of preservation would just be habitat preservation, pollinator preservation, or even possibly working towards full protection efforts. But I feel the most important thing to stress is education of the public, because as people become more aware of these issues, we can sort of work together to conserve not only Oenothera, but a variety of other plants and animals.
We would like to thank our mentors, Jeremie Fant, Matt Rhodes, Sadie Todd, Shelley Bender, Murphy Thomas, and our fellow college first intern, Marlene Ariano. And last, we would like to thank you, the crowd.
Yeah.
Do you think that has something to do with the cars coming in and possibly even bringing seeds from other areas? Do you think that's possible?
Well, since I don't think that has a really big effect because Oenothera is really dependent on its pollinators for cross-pollination. But we feel like the biggest thing that made them less inbred is just the spacing, because you saw on the map they were actually closer together. And it was just roughly 29 kilometers, and the hawkmoth can travel 30 kilometers each night. So it was able to travel back and forth between those populations. Any more questions?